with ligands and receptors, why do we need to measure the end result? And that's because if you are giving a drug to a patient, the patient doesn't care about the rate, about the binding efficiency, the affinity, the efficacy. They don't care about any of that. What they care about is, does the drug cause the effect that we are intending to happen? And so we have to have functional assays to see if the drug does what we want it to do. There are some pros and cons that go with functional assays as compared to binding assays. First of all, the functional assay answers the question, does it work? If we have a drug that has great affinity and seemingly has great efficacy at the receptor level, and it is non-toxic and it's cheap and it's all this stuff and it's a perfect drug, that doesn't do us any good if it doesn't actually cure the disease or treat the disease that we are trying to treat. Here we jump straight to the end and say, does this drug do what we want to do? Because if it doesn't, there's no use wasting time and money measuring all of these other things. We don't need radioactivity to do functional assays. We use radioactivity in a lot of our binding assays like we saw, but here we don't have to worry about any of that. And we have a lot of choice on how we're going to do our functional assays. We'll see that we can pick a number of different locations in the cellular cascade in order to choose what works best for us and what works best in our particular situation. One of the biggest drawbacks with functional assays, and we've talked about this in terms of the cellular response, is that all of our signals are amplified. And when you amplify things, you start to lose some detail, and we will see some of that. And because of that, we can lose some very important information that we can get from a binding assay, but we can't get from a functional assay. Here, we are looking at a graph that shows the relationship between the function and the binding of several different agonists. So when you look at this on the x-axis, each number represents a different agonist. So there are 6 agonists, and the names are up here, but what they are doesn't really matter to us. On the y-axis, they have maybe the most complicated unit ever, but just think of that as a measurement of the binding or the function. So if we look at agonists 1 and 2, in both of those, the white dot is higher than the black dot, and so it is more effective in binding and then producing a function. And so there's a loss of efficiency in converting a binding event to a response or a function event. Agonists 3 through 6 have higher function than binding, and we see that in general, as binding goes up, function goes up, but this is really it's unpredictable. You have to measure both for any agonist in order to figure out what the relationship is. So like I mentioned, when we choose a functional assay, we have a lot of choices in what it is we are going to test. And there's 2 main criteria that you're going to have to ask yourself in how and where we're going to test this. Number 1, what is the end result we're trying to measure? We want to measure the thing that's most closely related, the thing that's going to determine whether our drug treats a disease, but we also have to take into account what can we feasibly measure. Some things your lab may not be set up to measure, or maybe we just don't have the technology to measure the thing that you want to measure. And so in our figure here, we see that in this situation, we have 5 different things that we can measure. So in this situation, we have some sort of drug interacting with a receptor that's on the cellular membrane. And so we can measure receptor translocation. We'll see this later on in the lecture. But a lot of times when a drug or a ligand binds to a receptor, that receptor is actually going to move. Usually it's going to be internalized. It's going to go from the outside of the cell to the inside of the cell. Sometimes it may be, if the receptor is already in the cell, maybe it'll go into the nucleus, or maybe it'll go out to the membrane. We can look at the interaction of that receptor with other proteins. Think about our G protein coupled receptor. The receptor is sitting here on the membrane, but when it becomes activated, the G protein binds to it. And so we can measure the binding of that cytosolic G protein to the receptor. When that happens, we tend to produce other signaling molecules like cyclic AMP, and we'll see that in a little bit. Those are called second messengers. They're going to transmit the signal a little bit further down the line. And so we can measure second messenger production. The end result of a lot of different drugs and protein interactions is changes in gene expression. And so if we know that this ligand binding to this receptor should increase the expression of this gene, we can just measure the expression of that gene, and that the ultimate response to that gene expression is some sort of a cellular response, whether it is faster growth or death or slower growth or movement or whatever it may be. We can measure that thing at the very, very end of the line as well. So we always have to keep in mind, though, that the further down that line we go, we're going to get more and more amplification. And so we see that again here. So we have 3 graphs, and on each graph are 3 lines. Each line represents a different agonist. And so we're going to look at and compare the 3 agonists to each other depending on where we choose to make our functional assay. So our first assay is going to look at G protein activation, or you can think of it as activation of an ion channel. It is right after the ligand binding event. And so we see that all 3 compounds have different efficacies. 3 is most efficacious, 1 is least efficacious, and they have different potencies. While 3 is the most efficacious, it is actually the least potent. If we go a few steps down the line and we measure second messenger production, so think of this as like cyclic AMP production, now the efficacies are a little closer to each other. 3 is still the most efficacious, But 2 is very, very, very close, and 1 isn't that far behind, whereas initially 1 was way, way, way far, much lower than agonist number 3. If we go all the way to the end of the cascade, and we measure the cellular response or the organ response, now everything has been so amplified that all 3 agonists are capable of producing the same maximal response. Our potency has not changed though. And so fortunately, this amplification doesn't mask changes or differences in potency. However, it can make it very, very difficult to tell whether one drug is more efficacious than another, and also is something a partial agonist or a full agonist. So we're going to look more in detail at some different assays, and we're going to group them together based on where in that cascade we are looking. So we're going to have 3 different groups. Now, I am not incredibly concerned that you know which group number goes with which type of assay. What I need you to know is what the 3 groups are. And so for example, this one, group 1 is measure cells or tissue function. So you need to know that one of our 3 types of assays measures cell or tissue function. You don't have to know that that specifically is group 1. Group 1 assays are highly amplified. We're measuring at the very end of that cascade. And so because they are highly amplified, they are very, very sensitive. Something that a drug or an agonist that doesn't bind very well, has very low affinity, or when it binds has low efficacy, may be very difficult to detect or monitor if we use something that was less sensitive. So here, we take something that is undetectable and we amplify it up into something that is going to be detectable. However, because it's amplified, like we just saw, everything looks the same once you reach 100% response. And so partial agonists are going to look at agonists. And so, if you're using a group 1 assay, in this situation, we can't use it to say that this is a partial agonist and that is a full agonist. Because in reality, partial agonists and full agonists refer to their efficacy at the beginning of the line. How effective is that ligand or agonist at creating a change in the receptor? An example of a group 1 assay is microphysiometry. Now, I encourage you to go online and find some source so you can actually see this going on. I know there are some YouTube videos that look at it, and there's some nice pictures that you can find. But microphysiometry is a kind of interesting assay. In this situation, what we have is imagine a cell or some cells in a dish, and we are able to monitor in real time the environment around those cells or even that single cell. And by monitoring the environment, we can monitor the cellular metabolism. Every time a cell does something, whether it is to move or to grow or to express a gene, it requires energy. And if we require energy to do it, we're going to have to make ATP in order to carry out that process. And so the metabolism of that cell or those cells is going to increase. And so the goal of microphysiometry is to measure cellular metabolism as a measure of some sort of response to our agonist. And so we are going to measure 3 things in our environment. We're going to measure oxygen levels. If metabolism goes up, we're going to use oxygen and oxygen will drop. Same thing with glucose. If we make energy, we're going to reduce the glucose level. And we're also going to measure the pH. The pH is going to change as an effect, as a result of metabolism due to the production of lactic acid. And so as the cell metabolism occurs, the pH of that cell medium is going to drop. So we can measure that. And so at the top of our figure here, we see a basic idea of how this works. And so we're looking at 2 lines. On the x-axis, we have just time. And so we're measuring cellular metabolism as time goes along. The first line we see here is for some cells that do not express the receptor that our agonist binds to. And so we see that when we add this calcitonin, which is our agonist, we see no change in our metabolism. On the right, we have cells that express our calcitonin receptor. And so when we add calcitonin, we see a jump in metabolism. In the bottom left, what we're doing is we have some cells, and every time we reach an arrow, we are adding a higher concentration of our calcitonin. And so we see that every time we add calcitonin, our metabolism increases more and more and more. On the right, this is just a dose-response curve pulled from the one on the left, looking at what metabolism results from each concentration of calcitonin. Another way that we can do a group 1 assay is using melanophores. Now, I've mentioned melanophores before. We usually use melanophores from the xenopus. And xenopus is a species of frog. And melanophores make melanin, and the behavior of the melanin in those melanophores is dependent on G protein activation. In a melanophore, if the melanin is all clumped together, the vast majority of the surface of that cell, if you imagine looking down at a cell from the top, the vast majority of it is not going to have any melanin, because all the melanin is clumped together in one spot. And so that cell is going to look clear to our eyes. If that melanin disperses throughout the entire cell, the melanophore will look dark to our eyes. There are multiple G proteins. So we have G protein coupled receptor. We said when that becomes activated, the G protein binds to it. But there are actually different flavors of G protein. If we have a GI protein and that becomes activated, that is going to cause the melanin to clump together, making our cell clear. If we activate a GS or a GQ protein, that's going to cause our melanin to disperse, making it dark. And so we can get a couple of different parameters from this assay. Number one, we can find out, is our agonist activating a GI protein or a GS or GQ protein based on whether it causes our melanophores to get lighter or darker than where they start. And we can also use it to measure the amount of activation of that GI or GS, GQ protein. An agonist that makes the melanophore darker than another one is going to have more activity or more efficacy than the first one. So in our graph here, we see, both in graphical form and in just a picture form, the effects of agonists on these melanophores. So let's start with the pictures. You're looking at wells on a plate, and in those wells are melanophores. And so we can see, when we add albuterol or terbutaline, we are going to make those cells darker. And the more agonists that we add, the darker they are going to get. So it's a dose-response curve. And up above is simply the results from that plate down below graphed. And so you would use some sort of a spectrophotometer to simply measure how much light is getting through those melanophores. Shine a light through the bottom of the well and see how much comes out the top. So we see that both albuterol and terbutaline are capable of making 100% response, but albuterol is more potent. We start to see a response at a much lower concentration. We can see that both in the graph and in the pictures down below. These graphs are related. Again, we're looking at the ability of calcitonin to induce coloration in melanophores. And so in the left, we are looking at two different amounts of calcitonin. And so you can see that higher amounts of calcitonin cause a larger response in the melanophores than the lower one did. What's more important on this slide is the figure on the right. So in this figure, we are looking at dose response to calcitonin in melanophores, which is the curve on the left with the open circles, and in HEC293 or kidney cells on the right. So notice that these do not match up. We're looking at the same receptors with the same agonist in two different assays, and we get very different results. And so you have to remember that an EC50 or something like that is only pertinent to the specific assay that you are using it in. So I'm going to pause here. This is going to be the end of this video. The next video will start up here with our group 2 assays.